Genestack has been working with Rothamsted Research, a leading UK agriculture research institute, on integrating Ondex and KnetMiner agrigenomic suite of tools which enable network mining approach for gene-to-phenotype discovery. We have developed an application that builds knowledge networks by integrating multi-omics datasets including pathway, GWAS results, ontologies, homologies, and scientific publications. You can import your datasets using our smart importer, which automatically assigns the right data types to your imported files and lets you attach relevant metadata. To build a network, first select the relevant source files and later choose the NetBuilder app. This application lets you design the networks you wish to build. For example, the reference genome and the protein sequence are used to build a gene-protein network. This network is then extended using the protein annotation and gene ontologies to have additional concepts and relationships. Additional data sources can be added easily. These can include your private data, public data, or data from your collaborators. For instance, let's add a public GWAS dataset, which will introduce new relationships to our network, such as within genes and SNPs, as well as SNPs and traits. Once you've set up the data sources, you can start the computation that parse and integrate these datasets together into a network. When that's done, you can immediately explore the network using the KnetMiner application. This is the KnetMiner interface. When you search for a trait-related term, it will rapidly search the relations and concepts in the network to determine if direct or indirect links between genes and your searched term can be established. For example, let's enter a logical query, dormancy or anthocyanin. The result is a ranked list of candidate genes weighed by different types of evidence. For example, we see that this gene has a higher score than this gene, and the supporting evidence includes publication, biological processes, and so on. Users can then explore the results using a highly interactive network map. For example, here we can see that this gene encodes for this protein. And the protein happens to participate in the regulation of anthocyanin biological process. There is also some evidence from text mining. Here we can see that there is a co-occurrence relation between this gene and anthocyanin content from a particular publication. You can traverse the network further by revealing other relationships in the network. This type of network exploration is very useful for hypothesis generation. There is a bunch of other features, including the list of all evidences and a map of where the relevant genes are located in the genome. When you supply a list of QTLs, or when GWAS SNPs relevant to your search results are found, they will also be displayed in this map, and you can set the p-value association threshold. You can refine and increase the specificity of your search query using the query suggestions. You can also supply a list of QTLs to annotate and limit your search. Last but not least, you can also supply a list of genes to filter the search result. You can either enter this manually or by selecting genes from differential expression analysis results in the system. You can use homology relationships to combine networks between different organisms. This is useful to infer the functions of a less well-annotated organism. For example, let's construct a simple wheat network by linking it to a well-annotated Arabidopsis network using protein's homology relationship. For example, if we search for anthocyanin in this wheat network, we see that KnetMiner finds the wheat gene TRAS4AS to be relevant. This is because there are biological process and publication evidences for anthocyanin for the Arabidopsis protein AT3G29, which in turn is orthologous to the wheat protein trias4AS that's encoded by the gene trias4AS. We've seen how you can share data sources and the generated network, but in fact, you can also share your biological findings. The results that you generated are automatically saved, and you'll recover this view when you reopen this file or when you share this file with your colleague. The meta-info of each file keeps a record of how it's generated. It makes all the computation steps reproducible, and we can modify the data sources easily to update the network.